Hi, it's Dr. Ogden, and in this lecture we're going to be doing a brief introduction to cells. So cells are part of this microscopic world, and they're the basic building block of everything that we have in life. Now, the reason that cells are small, they need to be able to be that size so that materials can move around and get in and out of the cells with the speed necessary to complete the cell's metabolic needs. And everything that we've learned about cells has been accumulated in what we call cell theory. And the main two parts of cell theory is that all living things are composed of cells, and that all cells form from previously existing cells. Now, the study of cellular biology has been made possible with the use of microscopes. I mean, there are some things that we can see with the unaided eye, but other things we need microscopes. Like a light microscope lets us see down to the size of a cell and even inside of the cell. But if we want to get at the fine details of other structures, then we use an electron microscope. And with the electron microscope, we can see, down the structure of proteins and so forth. In fact, this is a picture of an electron scanning micrograph right here, and you can see on the outside of this cell all of these little proteins that are sticking out of it. So we've mentioned before that there are two types of cells: prokaryotic cells and eukaryotic cells. Eukaryotic cells are much larger and have a nucleus and then organelles, whereas prokaryotic cells are much smaller, do not have a nucleus, rather they have a nucleoid region and have very few organelles. Here's a quick kind of cartoon of a prokaryotic cell with some of the parts that it could have. So all of them have a plasma membrane and have this nucleoid region and ribosomes. But they also can have cell walls and capsules and pili and flagella as well. Whereas a eukaryotic cell, in this case an animal cell, again, is much larger. It also has the plasma membrane. It has the nucleus, which is a double-bound membrane, and then there are the organelles like endoplasmic reticulum, Golgi Apparatus, Mitochondria, centrioles, lysosomes, and flagella, the three of which are not found in plant cells, and there's a host of other things as well. A plant cell is very similar, has a nucleus and rough endoplasmic reticulum and smooth endoplasmic reticulum, Golgi apparatus, but it has chloroplasts, central vacuoles, and a cell wall, and these three things are not found in animal cells. So now we're going to look at the structure of a membrane, and it's made up of it's a bilayer construction where you have these phosphobilipid molecules here with a hydrophilic head and hydrophobic tails, and they arrange in this way where the heads go towards the liquid, so towards the outside of the cell, and the inside of the cell, which we call the cytoplasm, which is mostly water, and the tails face one another, right? Because they don't love water, hydrophobic, they're afraid of water. And this is the way that the cell membrane arranges itself. Now, that's a simplification, because in reality the cell membrane also has a bunch of proteins that are embedded within it that can traverse all of the way across the membrane or stick out on one side or on the other side. So you have the phospholipid bilayer and these proteins that are within it. And this is a fluid membrane. These molecules are constantly moving, and the proteins are moving as well. So why have all these proteins in the membrane? Well, they have lots of different functions. Here's just a few. Sometimes they're involved in attachment or in cell signaling and transport and enzymatic activity, intercellular joining, or cell-cell recognition. So if we look at now the different parts within the cell, we first are going to start with the nucleus. Now, the nucleus is where the DNA is held, and the nucleus is a double membrane, and that's called the nuclear envelope, and it has these pores within it, and we'll talk just a moment why we have these pores, but it's surrounded also by this rough endoplasmic reticulum. These little blue dots here are representing the ribosomes. So I want to just mention why we call the nucleus the control center, and that's because it has the DNA, which has all of the genetic information to tell the cell what it needs to do or to make whatever it needs to make. And the way that these messages are sent is a copy of the message is made in what we call messenger RNA or mRNA, and that is what leaves the nucleus. DNA never leaves the nucleus. That message comes outside, runs into a ribosome, and the ribosome is able to translate that message from a genetic message and turn it into an amino acid message. And eventually then can be built up, like we learned, into a long polymer of amino acids and become a protein. And so that's kind of the way that the cell controls what's happening, is through the DNA inside of the nucleus. And we'll learn more about that process later on. So, as I said, surrounding the cell you have the rough ER, and then you also have smooth ER. And the rough ER is simply called rough because it contains ribosomes, and so it looks rough. Here's an actual picture down here. And the smooth ER, rather, is where lipids are constructed. And if you imagine, here's one of these ribosomes, and here was a growing polypeptide that was made. That then can be butted off from the rough ER into this vesicle, and then it can be transported elsewhere. So it might come to the Golgi apparatus, and the Golgi apparatus's job is to work in partnership with the ER. It refines those products, stores them, and then ultimately distributes them. So it's kind of like the packaging company that sends the final product off to wherever it needs to go. Many times that might be outside of the cell, and sometimes it might be inside of the cell. Another organelle is called a lysosome. Lysosomes can have two main functions. The first function is that they help digest food, and so this is in the case like of an amoeba or something that brings in a big food particle, and a lysosome attaches to this food vacuole and eventually digests the food. Lysosomes also, though, you can take on the function of breaking down damaged or used organelles. So let's say that here's a mitochondria that's been used and used and used, and it's now ready to be recycled. The lysosome binds with it and can recycle all of those parts through the process of digestion as well. Vacuoles are found in both protists in the terms of contractile vacuoles, and then central vacuoles, which are found only in plants. And these are used, like in the central vacuole, it's used as a storage sac where they can store lots of water and other things, and that can contract and expand. Now we come to the chloroplasts, which again are, as we learned before, are derived from bacteria that lived symbiotically with these early eukaryotic cells, and they're the sites of photosynthesis, and we'll learn about that in another lecture. And they have a really interesting arrangement too, where you have an inner and outer membrane. You have these granum where are the sites where the photosynthesis actually takes place, and the whole point of the chloroplast then is to turn light energy into chemical energy. The mitochondria are the sites of cellular respiration, and so this is where you produce the energy source of the cell, which we call ATP. And this is always derived from food molecules or from sugar molecules. And so sugar is broken down, and the result is the energy source of the cell, ATP. And it also has an inner and outer membrane, and it has this inner folding. The next important thing in a cell are the cytoskeleton, and these are these microtubules that can stretch from one side of the cell to the other, and they're in charge of maintaining cell shape and also changing the cell shape. A good example of changing the cell shape is in the amoeba. Here you can see an amoeba as it's changing its shape, right? And this happens because the microtubules are extending out and allowing the cytoplasm to stream into those areas. Now, cilia and flagella are these motile appendages. So flagella are typically single, but there can be more than one flagella. Here's the case of a human sperm, where you have the head and then the tail over here, and the tail is this flagellum. In the case of a protist, you might have cilia covering the entire outside of the protist. Cilia can also line the insides of our bodies. For example, here are the cilia on the inside of a respiratory tract, and they move with a coordinated wave-like action, whereas the flagella are more like a propeller. So here's a quick movie of some cilia that are moving, and you can see how they're beating And that beating then causes a movement of the liquid. And so you can imagine how this might be used, to move things down or up the inside linings of organisms. So plants can also have these cell walls, and these cell walls provide the support, but they also can have these connections where you can have these channels between cells, and these are called plasmodesmata. Animal cells have similar things too, and we call these cell junctions. Sometimes they can have tight junctions or anchoring junctions, communicating junctions as well, and then this is all overlaid on the extracellular matrix. And so that's just a quick review of cells.